We're going to start with Watson and Crick and the double helix. And this is one of the most famous quotes from their paper, goodness, almost 70 years ago, where they published the structure of DNA, which is, of course, what this class is all about. Genetics. We're talking about DNA. What do they mean? Who can explain or interpret this quote? Give us a little bit more detail. What's the specific pairing they're talking about? Which is A's pair with T's and C's pair with G's, Chargaff's rule. So what does that have to do with how DNA is copied or replicated?
Does it make it easier for the enzymes to attach whatever nucleotide they would need?
Well, think about PCR. What's the first step in PCR? Right, you have a double-stranded molecule of DNA. You denature it. What's the next step in PCR? You anneal what? Primers. And then extension. So you've already seen DNA replication. What's the enzyme that does the extension phase? DNA polymerase. And how does it know? So let me draw in some base pairs here. Just a random sequence. How does DNA polymerase, sitting right here, know what nucleotide to add next? Right here. What's it going to put in there? A C, because that pairs with G. That's what Watson and Crick meant. This is way before anybody knew anything about the enzymes or primers or PCR or anything. They just said, this is the structure of DNA. We see the adenines pair with thymines and guanines pair with cytosines. That suggests a possible copying mechanism. That if you take a double helix and you split it in half, if you denature it, then how does that help us? Make two copies of the same piece of DNA. Each strand gives us the information, as we learned from Chargaff's rule, about what the nucleotides are going to be on the opposite strand. But before we got there, there was another famous experiment that proved, this is the Meselson Stahl experiment. You don't have to remember the names. Now, I've told you their names once, so you've heard it before. You don't have to memorize it. The Meselson Stahl experiment was the experiment that proved how DNA double helices are replicated. So that's the main take home for today, is to be able not only to understand how their experiment worked, but also to maybe apply it in new situations to really understand the basis for the experiment. And then to look at the molecular details of DNA replication. So that's where we're headed. Any questions at this point before we head in? Okay. So you've hopefully, if you read the chapter, or if you've taken high school biology or anything like that, you've seen a figure like this before, maybe. So we've got a double helix up here at the top. These on the left side and the right side are two different ways that DNA synthesis was thought could occur. So one of these two is true, one of them is not. And that's one of the questions on your Socratic exam. Oh, actually, I took that question out. Never mind. Sorry, you're not taking the wrong Socratic quiz. It's okay. So what's the difference between these two models? The conservative model on the left, semi-conservative on the right. Even if you don't know the details, just what do you see that's different?
In the conservative model, the DNA replicates, and you have one part in the first round of replication. One is going to be the complete copy of it, and the other one is going to be yes. And then on the other, the letter B, you're going to have one strand of DNA of the original, and the other strand is a copy. So you have both.
Right.
You have half of it.
Half original, half new. So the conservative model says you take a double helix. By the way, first question down here at the bottom. Why are the chromosomes colored differently? This is an important detail about Meselson Stahl's experiment. So yes, so you want to know which are the original strands of DNA and which ones are the newly synthesized strands of DNA. So that's the dark color are the original strands. And the light strands down here are the new strands, or the newly synthesized strands. How did Meselson and Stahl actually, they didn't color double helices, marker. What did they use to distinguish the parental strands from the new strands, or the original strands from the new strands? Yeah, so it has to be something chemical because we're talking about a deoxyribonucleic acid molecule. The original molecules, they studied this in bacteria. They grew the bacteria in heavy nitrogen, which is heavy, so hence darker color. Darker color, heavy nitrogen. Am I right so far? N15? N is normally 14. Thank goodness for the table of contents, periodic table of the elements. So they used heavy nitrogen. They grew the bacteria in heavy nitrogen, so all of their DNA in these bacteria were all heavier than they normally would be. They had nitrogen 15, not nitrogen 14. Those are the dark colored strands up there. And this is a type of an experiment called a pulse chase experiment. The pulse is you grow organisms in one sort of medium, like heavy nitrogen, and then you take those organisms out of the label, the pulse, so you pulse label with N15, and then there's a period of time where you chase, and all that means is that you put them back in normal medium, as normal nitrogen, N14. So that from that point forward, when you take the bacteria out of the heavy nitrogen and you put them in normal medium, N14, then all of the new DNA that gets synthesized after that point incorporates the lighter version of nitrogen. So up until this point in time, all of the molecules, all of the strands are dark because the bacteria have been growing in N15. And then in this first generation, what's the difference? We already heard the difference. We have, in conservative replication, you have one double helix. It's like you stuck it in a copy machine. You put that original double-stranded molecule of DNA in. You get out the original double helix. Both strands are heavy. And you get one double helix where the double helix has been totally synthesized anew from scratch. And that's the key difference between that and the semi-conservative, half-conservative. Denature the molecule in each of those two strands, the two dark strands here, there, and there, serve as templates for building the other strand of DNA. So second question down there at the bottom. What happens if you take those molecules of DNA that are produced by the second round of cell division and you centrifuge them? Centrifuging separates molecules by weight, mass. So N15 molecules are going to go farther down the centrifuge tube. Don't confuse this with gel electrophoresis. Similar principle, but heavy. Draw, make a little test tube on your slide for both of those two models and draw out the patterns of bands that you'd expect from those molecules of DNA.
Are you centrifuge all 8 in the same one, or like?
All 4. So it's either the 4 on the left circled in black.
Or the 4 lines, so separate.
Yep, so 2 tubes. You combine all 4 of those. So what are the different patterns you're going to get? How many different bands are there going to be on the left side? How many different sets of nucleic acids? 2. Okay, one is totally heavy, 2 heavy strands, 2 dark molecules. So that's going to go down there to the bottom of the tube. And then the other type is, we've got 3 molecules of all light. So they're going to be higher up in the centrifuge tube. And this was how Meselson and Stahl could distinguish these two possibilities, is that the pattern, hopefully you've gotten to the pattern on the right, how does it differ? What's the difference between the left side and the right side?
You only have one original strand.
Instead of both of those two original strands being part of the same molecule and being an all-heavy band, you've got the half-heavy bands, except I can't spell, half-heavy, and then two of those. And then you've got two molecules that are light.
Question.
Yeah,
I mean, I feel like the semi-conservative model would make sense, because if polymerase is gonna, you denature your DNA, and then polymerase comes in and it attaches.
yep,
So
but
you
they
have-
didn't know that at the time.
So now it's.
Now it makes sense. When I taught this class last semester, I tried to explain, because I always start class with, as we know, semi-conservative replication is what happens. You melt the two strands, and then DNA polymerase uses each strand as a template to build the other strand. I can't even come up with, and maybe one of you can help me, I can't come up with how to explain how this would happen. How you'd take, how would you have an enzyme that would take a whole double helix and spit out an original and a total copy?
Unless it involved more denaturation right at the end.
I-
But that would still not be replication. You'd have to denature and reassemble them differently.
Right.
It seems like it use it like a copy machine, where you make a copy of something.
Yeah.
And then you still have your original copy, but you have this new one.
So that could have been just an alternative. Like, I think I said copy machine actually today, didn't I? So you could imagine an enzyme that has a big hole in it, you feed in a double helix, it reads the double helix and it synthesizes a new double helix. That was what they were trying to distinguish here. And it turns out, so N15, double helix, will wind up at the bottom of a centrifuge tube. If the top's up here and the centrifugal force is down, N15 molecules will go down to the bottom of the tube. The lighter N14 would float a little bit more. They won't get pushed as far down the centrifuge tube. So N15, I guess I should use some sort of color coding. That would be dark molecules, all light, so that's all heavy. What happens to those intermediate molecules? That should be pretty straightforward. What happens if you have a molecule that's, as was predicted in some of those, one light strand, one heavy strand? It's in between. So there's a possibility of getting three distinct regions of this liquid in the centrifuge tube. Liquid in the centrifuge tube. And we're going to find out, based on whether or not conservative or semi-conservative replication happens, what the pattern of bands in this tube is. Yeah.
If we talk about DNA replication,
We are talking about DNA replication. Yeah?
if you replicate DNA, it's supposed to be the same, because we're using the same one from, that it's a same copy, like the daughter should be the same as the first one. Why then it would be light or heavy if they are like the same?
Same nucleotide sequence, different nitrogen atoms. So the nucleotides are the same. A's, T's, G's, and C's. The sequence is the same. The only difference is that, and this is the reason that they use nitrogen instead of, well, carbon would have been useful too. I was trying to think of an element that's not in a nucleotide. The point is that nitrogen is in nucleic acids, nitrogenous base. So it was an element they could use that would label every one of the four different bases, A's, T's, G's, and C's, and would let them track which one was an old or pulse-labeled strand, and which strand was a new or light strand. So the sequence of the bases doesn't differ. The only difference is here black has N15, and the blue strands are containing light versions of nitrogen. Sequence of the two strands is the same in all of these cases. That this is what Meselson and Stahl saw. The actual figure from their publication so many years ago, 1958. They had cameras back then? So here's what it looks like. We've got on the right there the three molecular weights of the bands. Light at the top, then half heavy, then heavy at the bottom. So they're not spread out as far as they were in my drawings, but you can see there are three different series of bands. The weird thing about this is that time's going from right to left. So each of these strips is they waited a certain period of time with their bacteria growing in their tissue or their culture flasks. They'd pull a little bit out, and then they'd look at the molecular weights of the DNA. And they'd wait a little bit longer. So this first reading was at 0.3 generations. So their unit of time is how many times have the bacteria replicated or divided. So here's after the first generation of bacterial growth. What's the only band there? All of the DNA is half heavy after one generation. And after 2 generations, 1.9 generations, I'm being generous and rounding here at the top, 2 generations. Half of the strands are heavy, light, and half of the strands are half heavy. So what's the overall pattern? What's happening as generation proceeds this direction? It's semi-conservative replication, but how would you describe this pattern of bands?
Higher percentage of nitrogen 14 base DNA.
You're getting more and more N14 or light DNA, which is not surprising because you only fed those bacteria N15 for a short little period of time. So you give them a tiny bit of heavy nitrogen. And as time proceeds, what happens to that heavy nitrogen? N15. It's getting diluted. Every generation that DNA synthesis occurs again, you're incorporating more N14 into all of the DNA and all of these bacteria you're growing. What happens to the N15? The heavy nitrogen that was originally there. Is it still there in the 4th generation?
It's there.
It's there, it's not prominent.
Right, so there's still a half heavy band there. It's just really light compared to all of the new DNA that's been synthesized, that light band, this super thick, dark band up there. So what happens eventually? The bottom question. After generations equals infinity, what's the outcome of this experiment?
It'll be there, but it just probably won't be detectable at that point.
There are in this, yeah, exactly. It won't be. It is interesting about this experiment is when I started thinking about the implications of this. So what does it mean that those original 2 strands of DNA are still present in this bacterial population after an infinite number of generations? It's weird, I admit, but what I think of is maybe those 2 original double helices that made me are still in my body somewhere. Maybe they're in my toe, I don't know. Right? Semi-conservative replication. The original strands of DNA that made us, single cell embryo, those 2 strands don't get broken and reassembled and shot into different parts of our bodies, different cells. They stay intact, and they stay in us, serving as molecules to replicate more DNA and more DNA and more DNA. It's like a family tree of our DNA. Single cell embryo spawns all of the DNA molecules that exist and get synthesized during each round of cell division as we grow and develop. And all organisms, not just us, of course.
Dr. Ross?
Yeah.
So I know we talked about the conservative and semi-conservative, and the book also mentioned
Distributed.
Or yeah, is that more kind of based off of like what happens in the meiosis? That theory more so than what's crossing over kind of thing.
I opt not to talk about distributed DNA replication for a reason because I don't want to get confused with what happens during recombination and meiosis. And so if you read, and I hope you did, just eliminate distributed replication from your brains. It was another alternative hypothesis about how DNA replication could occur, but Meselson and Stahl didn't test that. Well, this did test that hypothesis. They didn't explicitly test that hypothesis. And yeah, I don't want to get confused with recombination, which seems related to distributed synthesis.